students, welcome to Dr. Deepa Dahake's NEET Biology, and in today's session we study about another keynote for NEET 2020 exam from class 11th. In today's session, as you appear on the board, we mainly discuss about the anatomy or internal structure of the muscles fiber. Topic is under Locomotion and Movement chapter of human physiology. When we come towards the muscles fiber, so here you remember we mainly study about the skeletal muscle structure, because the skeletal muscles are mainly responsible for the locomotion in humans. Now, when we come towards the structure of muscles fiber, most important topic for your NEET exam, because the number of difficulty level questions are asked from these structure. So here when we start with the structure of or anatomy of muscles fiber. So inside the body, you remember how these structures are present, that I explain in a very short way. First you see the bundles of muscles are present in our body. These muscles bundles are commonly called as fascicles. Every bundle or every muscle bundle, you remember, consists of large number of bundle fibers, and further every fiber is made up of, this part is important, every bundle fiber is made up of parallelly arranged filaments, and these parallelly arranged filaments are called myofilaments, or they are called myofibrils. When we come towards the structure of skeletal muscles, so predominantly one thing you remember, we mainly study about the structure of this myofibril or the structure of myofilament. So here now we discuss about the structure of myofilament or myofibril. Every myofilament or myofibril, you remember, is made up of alternate bands, that is, one is dark band and adjacent to dark band immediately light bands are there. So you simply remember the myofibril filament is made up of alternate dark and light bands. When you come towards the objective section, so instead of dark and light, question is asking in the form of A band and I band. And mostly students get confused between the A and I band. That's why you simply remember the dark band, A underline kariye. The dark band is called as A band, and the light band, again the second letter you underline I. So light band is called as I band. Now why these bands are called A and I? Reason is the A band is anisotropic band. Why it is anisotropic? Because the thickening of or the thickness of A band, because the thickness of A band is universally not similar. Somewhere the A bands are more thick and nearly in the centers A bands are shows the lighter region. That's why they are called as anisotropic band. When we come towards the I band, throughout the length of muscle fiber, wherever I bands are present, all the I bands are showing nearly similar thickness, and that's why they are called as isotropic band. Now, the A bands, what are the proteins? The main question. The A bands and I bands are made up of what kind of protein? So, A band, you remember, mainly made up of thick, because it is darker in appearance. So, it is made up of thick proteinaceous filament. I band is lighter in appearance. That's why they are made up of thin filament. Thick filaments are made up of myosin. You keep in mind very well. The thick filaments are made up of myosin protein, and the thin filaments are made up of actin protein. So this thing you remember. The dark bands are made up of myosin, and the I or light bands are made up of actin protein, that is forming a thin filament. Now this part is most important, that in every dark band, you remember, in every dark band or A band, centrally somewhat lighter zone is present, that lighter zone is called as H zone or it is called as Hansen zone. And one of the most important question for your NEET exam, the zone of the muscle fiber or the part of the muscle fiber which disappear during muscle contraction. So this you remember, the H zone is mainly disappears during the process of muscles contraction. So this you remember. And what is H zone? H zone is present in the center of the dark band and it is somewhat lighter than the peripheral region of the A band or the dark band. Now this is the theoretically we study this part you remember. Now we come towards the diagrammatic section for the same whatever we study. When we appear under a normal microscope, so here you remember this kind of structures are appear. The dark band followed by light band and the light band is followed by dark band. That means alternate pattern is there for the dark and light band, or you can say for the A and I bands. Now what here this diagram is showing the detailed portion of one dark band and two I bands. So here you remember this is one complete dark band is there and two surrounding sides Z lines are appear clearly. So these two Z lines are showing the I band. When we come towards the A band, so this A band you remember exactly in the center of the A band. One vertical line is appear. This line you remember it is called as M line. And the filament of myosin, M line as well as M for myosin also you remember. The thick filaments of myosin here only in one filament I darken the zone. So the thick filament of myosin you remember they get attached or they always bind with the M line. And because of that you remember for objective portion most important thing that the thick filament of myosin never change their position. Only when you study about the sliding filament theory, so the thin filaments are slide over the thick filaments of the myosin because these thick filament of myosin are connected by the centrally located M line of the A band. Now if you appear this A band towards both side of the M line, then here you clearly appear this much zone is appear very dark in microscope, but nearly towards the both side of the M line or nearly in the center of the A band, one H zone is present. Why the H zone is slightly or somewhat lighter? H zone, first thing you remember, we only appear this H zone during the relaxation of muscles. When the muscles or muscle fiber are in relaxed state, then we appear this H zone in the muscle fiber. Why we appear? Because during relaxation only the A band shows the dark filament or it shows the myosin filament. The thin red color से आपको appear हो रहे हैं. The red colored filament here we show that the thin filaments of actins are not present in the center of the A band. They are only present at the parallel surface or at the outer surface of the A band, and because of that the A band shows the H zone, and exactly in the center of the H zone you remember M line is present. When the muscles start to contract means the Z lines from both the side slowly come towards the center. When Z lines start to come towards the center of A band, so the actin filament or thin filament which are bind to Z line or joint with Z line, these filament gradually slowly start to come towards the center, and because of that the H zone is disappears. So this you remember. Z line you remember. Z line responsible for equal division of I band or light band. So isotropic band equally divided into two halves due to the presence of Z line. Now what is sarcomere? Another question. Sarcomere you remember. What is sarcomere? the area between two successive Z lines is called as sarcomere. And how much area it covers? For that also question is generally asked. So sarcomere is consist of this, you remember, sarcomere is consist of a single A band or one complete A band and two half I band. This part you remember very well, the two half I bands and one complete A band combine to form sarcomere. What is sarcomere? Sarcomere is a unit of structural and functional unit of muscle contraction. So this you remember, sarcomere, the structural and functional unit of muscle contraction. During muscle contraction, you remember, the length of filament never be changed. Only the area for sarcomere is decreases because the H zone get disappeared during contraction. And then one term is there, sarcoplasm. Sarcoplasm means, you remember, the protoplasm or the cytoplasmic part of the muscle fiber is called sarcoplasm. And one another term is repeatedly asked in exam, that is sarcoplasmic reticulum. Sarcoplasmic reticulum term we use for the endoplasmic reticulum which is present in the protoplasm or cytoplasm of the muscles fiber. This part you remember. One term is provided in NCERT, that the sarcoplasm is syncytial. Syncytial means, you remember, it is multinucleate in nature. So this much you remember for the structure. This diagram you keep in mind, very well you keep in mind. Okay, now when we come towards the myosin and actin, so one another question is raised, that what are the proteins? So now is it clear? The dark band is made up of myosin filament and thin band is made up of actin filament. But when we study these proteinaceous part in detail, so here you remember, for now we come towards the proteinaceous part of the muscles fiber. Predominantly we say the two proteins are there, but in addition to myosin and actin, tropomyosine and troponine are the another two proteins which also involve or participate in the formation of thin filament. First हम study करते हैं. First we come towards the thick filamentous protein or the protein which is responsible for the formation of thick filament, that is the myosin. When we study about myosin, so here you remember, myosin is formed. Myosin is a polymeric protein or it is a polymer. Clear? And this polymer is made up of large number of monomeric unit. What is the monomer? Monomer, you remember, Meromyosine units are the monomer of myosin protein. Now, when we come towards the structure, so this is the single monomer. It is not a entire myosin. This is a single meromyosine unit, and this meromyosine is made up of two parts. The head part forms the heavy meromyosine, and cross arms and tail, mainly tail, forms the low meromyosine. Tail as usual made up of hydrocarbon. Head, again the question is asking for these two sides, that the ATP binding side, ATPase activity side and actin binding side. So the thick filament having a binding side for thin filament, and thin filament equally show the binding side for thick filament. So here we study about the binding sites for actin. So that over the head in both the dimer के form में, so in both the anterior end of the head, you remember one actin binding site is there, and literally ATPase site or ATP binding site is there. You can say ATP binding site or you can say ATPase site, because ATPase enzyme is present here, and at the time of muscle contraction ATP is required, so that ATP is present at these sites of the myosin protein. Now such number of thousands of the meromyosine units combines with each other. Combines means parallelly arranged with each other, and they will result in the formation of one thick filament that is called as myosin. So meromyosine, this is the structure for meromyosine. For NEET exam, you remember literally placed ATPase side and anteriorly placed this is the actin binding side. And when this actin binding side of the myosin bind to actin, so it not directly bind to actin, it always bind with actin via the calcium ion. So calcium ions also equally play a significant role in binding of myosin and actin filament. So this you remember. And now we come towards the thin filament. What is thin filament? Thin filament, normally what we are studying, that the thin filament is made up of actin protein. But not only actin. In addition to actin, the two another protein, tropomyosin and troponine, are also participate in the formation of thin filament. But the framework of thin filament is formed by actin. That's why we say the thin filaments are made up of actin. Now, under resting condition or under relaxed condition, you remember the actin is present in G-actin. G-actin means it is present in globular form. So the globular actin के form में present है, and this globular actin you remember when convert into fibrous actin, then the actin become in active state and this form is called as F-actin. So actin you remember predominantly present in two forms. One is G-actin and second is F-actin. Normally during contraction, whenever we draw these thin filaments, that means we draw the F-actin. Now this F-actin is made up of two parallelly coiled filaments. So two parallelly coiled filaments सबको appear हो रहे हैं. And after that you remember the second is tropomyosin. Smaller subunits हैं tropomyosin proteins, and these tropomyosin, again these red color part appear. So this tropomyosin is bind over the F-actin, and then with this tropomyosin the another trimer is bind, that is called as troponine. In NCERT, 3 units are not provided in the structure part, but their role is provided when you study about the mechanism of muscle contraction or you study about the sliding filament theory. So here you remember troponine is mainly made up of 3 units, TPC, TPT and TPI. TPC means the unit by which or through which calcium ions are bind, and with that calcium ion the actin binding site comes in contact. So this part you remember, because during contraction actin filament is slide over the myosin filament. After that, or this TPC you can say involve in cross bridge formation. Cross bridges are formed during the contraction between actin and myosin filament. TPT, T here stand for tropomyosin. So this troponine unit bind with tropomyosin with the help of this T. And inhibitory unit is represented by I. When the muscle contraction stops or completed and muscles start to become enter in relaxed condition or repolarized state में enter होते हैं, at that time you remember instead of calcium binding site, the inhibitory sites become active and they mask these calcium binding sites. So maybe कुछ स्टूडेंट्स को जिनका कॉन्सेप्ट पार्ट वीक होगा उनको कुछ पार्ट यहाँ पे स्किप लगेगा या नहीं समझ में आएगा, बिकॉज वन फुल लेक्चर इज रिक्वायर्ड टू अंडरस्टैंड all these 3 subunit. But for revision purpose and keynote purpose, you remember the thin filament is made up of actin, tropomyosin and troponine. Troponine bind to tropomyosin and this tropomyosin is bind with actin. Myosin is made up of single subunits or monomeric unit, and this is not the structure of myosin. This is the structure of single unit of myosin, that is meromyosine. So simply some most important thing you remember, the myosin when bind to actin, so both are linked together by calcium ions. That calcium ion is bind with the TPC unit, and this unit one side bind to the actin binding side. So this part you remember, myosin show actin binding side, actin show myosin binding sides. The part which is disappear during muscle contraction, again the H zone of the muscles fiber or sarcomere. H zone is disappeared during muscle contraction. Which of the following part or length, true and false type of question you search for this topic? So you appear one question regularly and repeatedly आ रहे हैं वो question, and question is related with the shortening of the fibers and shortening of the which of the following undergoes in shortening phenomena. So here you remember the fibers never be undergoes in shortening. Shortening of only sarcomere or the area of sarcomere only reduces during the process of muscle contraction. So I hope somewhat I try to cover in the form of keynote, तो वो part आपका clear हुआ होगा. And even after that again कह रहे हैं कि कहीं doubts and queries है तो आप doubts को clear कर सकते हैं पूछ के. Clear? In next session we study about the nerve impulse conduction, because after this locomotion and movement chapter deals with the skeletal system and how does the impulse conduction is take place. So we mainly in next chapter study about the next topic, we study about the nerve impulse conduction for NEET 2020. And up to next session, stay connected, work hard and stay blessed.